21. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 missing. I've just started marking them because I just finished the photosynthesis gizmos this morning. So I've just started marking the other blocks. Okay, I guess we can get going. So what we're looking at today is this one here, May 31st to June 4th, and we're going to be looking at protein synthesis. So going from genes to proteins. Um, this one's a long one. This will probably take 2 days to cover, like 2.5 days, because it's like 43 slides total. This is going to take a bit of time to get through. Okay. Oh, it's over there. So I guess we can get started. So basically, we're going to talk about how genes work. Okay. Now, what do genes code for? So the basic questions: how does DNA code for cells and bodies? How are the cells and bodies made from the instructions in DNA? That's what we're basically looking at in this PowerPoint. So we start with DNA. Okay. That's going to make proteins. Those proteins then will function together in cells, which basically make up our bodies and our physical traits. Yeah, no worries. So remember yeah, after school. Okay, so we have the central dogma right now, which is what Watson and Crick basically came up with. So it's the flow of genetic information in the cell that we've discussed before. Okay, so how do we move information from DNA to proteins? Okay, bye. You have DNA. Okay, Neela. And DNA can make copies of itself, which we call replication. Okay, and then we can also produce RNA, which is transcription, and then that RNA will be used to make proteins, which is translation. That produces a trait, which is basically the phenotype. The question I pose to you is, why go through this extra RNA step? Why not simply go directly from DNA to protein? Okay, that's the question we're pondering right now. Now, where do we normally keep DNA in a cell? Okay, so it's in the nucleus. Okay, and where in the cell are proteins made? Where are the ribosomes located? Yeah, in the cytoplasm. Okay. Why do you think we never want DNA out in the cytoplasm? Well, let's approach it from another point of view. I'm assuming most of you had lunch today. No, it's not permeability. Okay. Can we say with pretty good confidence, yeah, okay, Grant's on the right track, that the cells that you ate for lunch today, be it animal or plant, would contain nuclear material? Yeah, okay. That nuclear material is going to end up, you know, inside your different cells. But it doesn't remain intact. It couldn't, because no worries. If your DNA, if let's say you ate a carrot and the carrot's DNA stayed intact in your cells, you would start expressing carrot genes, which as a human being probably doesn't make too much sense. Okay. So what are your cells going to try and going? Your cells want to use the nucleotides in that DNA of the carrot or the celery or the beef or the chicken. So what is are your cells going to do to that intact DNA that's now inside your cytoplasm? Yeah, it wants to break it up. How's it going to break it down? What's it going to use to break it down? Okay, enzymes. So the specific enzymes that your cell uses to break down nucleic material are called, and let me click on the chat so that I can put properly, nucleases. Okay. Now, the problem with nucleases is that they cannot differentiate between the DNA from you. Okay. Okay. The DNA from you from the DNA in a carrot or a celery or a chicken, right? So these enzymes are simply going to break up long strands of nuclear material. So if we had DNA from your nucleus, leave the nucleus and try to get to the protein in order to, or to the ribosome in order to make a protein, the DNA is going to get destroyed. Okay. So if it gets destroyed, what happens then? What good is it as a genetic information storage mechanism? There isn't any usefulness at all. So this is why we have this one extra step. By copying the DNA to RNA, the DNA stays safe inside the nucleus. It never gets out. The RNA can leave, and the RNA can go out to the ribosome carrying a copy of the DNA message, and the ribosomes will then make the protein which makes the trait. And it doesn't matter if the nucleases then destroy the RNA, because you don't want to make the protein forever anyways. So long as the signal, the RNA, sticks around long enough to make sufficient protein that you need, it doesn't matter if the RNA gets destroyed afterwards. Okay? The problem with going DNA to DNA has to do with the fact that you have to be able to differentiate in terms of what can be destroyed and what can't, right? So if you make something different, it's easy to say, oh, okay. It's easy to pick out that one. That's the one that has to be destroyed. Yeah, that's why foreign RNA can be used, right? That's why the mRNA viruses work fairly well. We no longer need to use living or dead viruses. We can just use the mRNA from it, right, to express whatever DNA that we want. It theoretically could be, theoretically. But remember, to be permanent, right, to be a permanent change, the RNA would have to be transcribed back to DNA, and then it has to get into the nucleus and replicate with the rest of the host DNA. So DNA is a better long-term storage mechanism. The other aspect, right, structurally, the RNA is different from DNA. RNA is much less stable and more susceptible to mutations. And therefore is easier to screw up. Okay, so metabolism taught us about genes. Okay, so if you look at the inheritance of metabolic diseases, things like Tay-Sachs disease, PKU, albinism, diabetes, they're all due to the disease, which is a certain type of phenotype caused by a non-functioning gene product. In diabetes, you're not making insulin properly. In albinism, you're not making melanin properly. In Tay-Sachs, you're not making a fat dissolving digesting enzyme properly, so you get abnormal fat development in the neurons of the brain, and the kid's usually dead by about age 5. PKU is another similar idea. So we know in metabolism, we have a metabolic pathway. If a mutation occurs at any one of these points where these enzymes are, so let's say enzyme 1 gets knocked out, then you get a disease. Okay. If enzyme 2 gets knocked out, you get a related disease because it's the same metabolic pathway. Same with enzyme 3 and enzyme 4. So this would be a family of metabolic diseases that would be related to each other because one enzyme within this pathway has been knocked out. Okay, so this whole idea was basically put together by two scientists by the name of Beadle and Tatum. And the idea of their hypothesis was called the one gene, one enzyme hypothesis. And they won Nobel Prize in '58 in medicine for their discovery. All right, so let's look at their experiment. Okay, so the organism that they chose to work on is bread mold. So that bread mold's name is Neurospora. Okay, and if you look at bread mold, you'll often see that it's in multiple different colors. It's the black stuff. Okay, that particular fungus is the one they decided to work on. Okay, so what they first did, they captured Neurospora from the air because the spores from bread mold are just floating around in the air. You don't need to do anything special to capture them. You just take a glass and whip it through the air like this. You're going to grab a handful of Neurospora spores. Okay, now in this test tube that they captured these funguses in is something called minimum media. Minimum media is the basic stuff that you need, the minimum requirements that you need in order to grow something, right? So it contains nothing complex. So no vitamins, no complex carbohydrates, nothing like that. Only simple sugars, right? Maybe some fatty acids, the minimum requirements to keep something alive. Okay, and they put the wild type Neurospora into this mixture, and the neurospora grew, okay, which told them, great, we got some neurospora. Okay, they then, when these neurospora reproduce, they produce spores. So what they did was they hit these spores with either X-rays or ultraviolet light. And the purpose of this was to induce mutations in these spores. Okay? Now, once these spores were irradiated, they got transferred to a different test tube. And in this test tube, the growth medium contained complete medium. This means this mixture contained every single complex component that the organism needs to survive. So these spores do not need to make anything. Everything they need to grow is supplied to them. Okay? Why? What are they checking for? This is a control. In fact, it's a positive control. Why would they, after radiating the spores, check that the spores could grow if everything is provided to them? What's the point of that control? What's testing for? Nope. What does mean if you hit a cell with too much radiation? What happens to that cell? More than mutations. Yeah, it'll die. Okay. So the purpose of this test was to see, did we hit it with too much radiation or not? So the point was to make sure that these cells weren't dead. Okay. Once they knew that they could grow and they were not dead, okay, now you have to try and find out if a mutation occurred. So they took this one of the spores, because they're only looking for one mutation, and they grew that one spore on complete media, okay, and then took a sample of those and tried to grow them on minimum media. So this is a negative control, okay? Since the spore will grow if you give it everything, but the spore doesn't grow when you give it just the bare minimum, that must mean that there's a mutation. This fungus can't make something that it needs and therefore cannot grow if everything isn't provided. So this confirms the presence of a mutation. Yeah. Okay. So now the question is, how do we figure out what the mutation was? So these are all the experimentals. Okay. So what we're going to do, we're going to take samples from this test tube and we're going to put it on minimum media plus one additional supplement. In this case, pyridoxine. In the next one, p-aminobenzoic acid. Then choline, inositol, nucleic acid, folic acid, arginine, niacin, riboflavin, thiamine. Okay, so you're going to give one nutrient plus the minimum media. Okay, so those are the experimentals. If you look at all of these test tubes, yeah, after school. Okay, after so within all the experimentals, which is the only group that shows growth? Yeah, this one here. It's the only one with a little bit of orange here. Okay, so these are the supplements, and this is the only one that grew, which means the mutation that we caused somehow broke the pathway to make arginine, right? If you gave it niacin, it doesn't matter. It can make niacin. It's not missing niacin. It doesn't matter if you give it thiamine or nucleic acids or choline. What it can't make on its own is arginine. Once you give it arginine, it starts growing. Okay? All right. So we've now identified the mutation caused up here as being one that's preventing the formation of arginine. Okay. So Beadle and Tatum realized that to make arginine requires a number of steps. Each one of those steps is controlled by an enzyme. Okay. If that enzyme is mutated, then the production of arginine stops. Okay. So here's the pathway that they worked out. You start with some amino acid. Okay. Then enzyme 1 will convert it to ornithine. Enzyme 2 converts it to citrulline. Enzyme 3 converts it to arginosuccinate. And enzyme 4 converts that to arginine, and the fungus uses arginine to grow. Okay. If the radiation broke gene E, enzyme 1 isn't produced. If enzyme 1 isn't produced, none of these steps can occur. Arginine isn't made, and the fungus can't grow. If enzyme 2 is the one that's busted, then again, same result. Arginine can't grow, or enzyme 3 or enzyme 4. So the question was, how do you determine where the broken gene was? Well, it turns out they broke 4 genes, depending on which experiment they did. When they broke enzyme 1, the precursor would build up inside the fungus, and there would be none of these produced. If enzyme 2 was the one that was busted, then ornithine levels would go up and everything else would remain low. If it was enzyme 3 that was broken, then citrulline would go up. And if enzyme 4 was busted, everything would work up to arginosuccinate, and then arginosuccinate would build up and arginine would not be made. So they could actually tell where they actually broke the pathway. Okay. From this, they made an argument for what they called the one gene, one enzyme hypothesis. Basically, that was saying that every single enzyme was coded for by a single gene. Okay, so if you have 4 different enzymes, you need 4 different genes to make that metabolic pathway. Okay, now that was later modified because are enzymes the only things that proteins make? Mm-mm. Look at your hair. It has right. Your hair has keratin in it, which is a protein. You've got insulin, which is a protein, and that's a hormone. You've got collagen, which is used for structure in your skin. Those are all proteins. So this was later changed from the one gene, one enzyme to the one gene, one polypeptide, saying that every single polypeptide in your body had to have a gene that created it. Okay, so that was that hypothesis. That's what they came up with, basically. Okay, so now let's take a look at this process. Okay. So the first thing we're going to go talk about is transcription, which is basically switching from a nucleic acid language to a different nucleic acid language. So it's like going from speaking to someone who is from Newfoundland and trying to get them to speak to someone from Vancouver. Right? They're both speaking English, same language, but they won't necessarily understand each other. Okay. Anyone here from Newfoundland? Or you went there? Okay. Yeah. No. It's really interesting, especially if you listen to an accent like an accent from Stevensville. Oh, you think that's weird? Go to Stevensville, Newfoundland. It's the weirdest accent you will hear. They sound Quebecois. Okay. So if we look at DNA versus RNA, okay, the big difference is the sugar, right? So RNA uses the ribose sugar, DNA uses deoxyribose. Okay, the nitrogen bases are different. Okay, so RNA uses uracil instead of using thymine. Okay, so the pairing is U to A and G to C. Okay, that's one key point. People screw up all the time. Is they keep putting T's instead of U's when we're talking about RNA. So please keep that in mind. Okay, RNA is single-stranded, which is why it mutates more easily and is less stable. With DNA, right, you can double check. With RNA, you can't. Okay, and then another big difference, there's really only two kinds of DNA called alpha and beta, and that just kind of depends on the shape of their twist. Whereas RNAs, there are a lot of different kinds of RNAs. There's mRNA, tRNA, rRNA, siRNA, SNRPs, and all kinds of things. SNPs, SNRPs, there are other RNAs. Okay, so let's talk about transcription. All right, so how do we go about making messenger RNA? Okay, the side of the DNA strand that we copy from, okay, the side that is transcribed is called the template strand. The side that we are not copying is called the coding strand. It ultimately has the same sequence as the RNA. So look at this. In green here is the RNA that's coming off, and up here is the side that's not copied, right? The only difference between the sequence of DNA here and the sequence of DNA here, or sequence of nucleotides here and here, is that the T's are replaced by U. Otherwise, they're exactly the same. TU, GG, CC, TU, AA, CC, GG, AA, and so on, right? So the mRNA code will exactly match the side that is not copied, which is why the side that is not copied is called the coding strand. The side that is copied is the template strand. Okay? The structure that is currently unwinding and making the complementary strand is called a transcription bubble. Okay? And the enzyme that does the transcribing is RNA polymerase. Okay, so this is a template strand, and here's the coding strand. Okay, because this is a polymerase, like all polymerases, RNA polymerase also builds the new RNA strand 5' to 3'. Okay, also note, this is a much simpler mechanism than DNA replication. DNA replication, you've got like 6 enzymes and binding proteins involved, right? Ligase, poly 1, poly 3, all these different components. In transcription, you'll use one enzyme, and that's it. You don't even need to worry about single-stranded binding proteins because the enzyme is big enough that it can hold the transcription bubble apart on its own. It doesn't need SSBPs to stop annealing. And then as it leaves, it rewinds back up. Okay? All right, the RNA polymerases. There are 3 RNA polymerases. RNA poly 1 makes rRNA. We'll talk about what those are later. So it's involved in ribosome production. RNA poly 2, which is the one we were just talking about, is the one that transcribes DNA genes into messenger RNA. And then RNA poly 3 makes something called tRNA genes. Okay. How does the polymerase know what to make when? Poly 1 only recognizes a specific sequence that will allow it to make ribosomes. Poly 2 only recognizes a specific sequence that will allow it to translate messenger RNA. And poly 3 only recognizes a specific sequence that will allow it to make tRNAs. Okay, so Poly 3 cannot go to where Poly 2 normally is and start transcribing. It wouldn't know where to start because it would not recognize the starter position for Poly 2. Okay, so each one is specialized into functioning or starting only at its own sequence. Okay, so how do we know which is the template strand and which is the coding strand when we have a chunk of DNA? Okay, well, the DNA that's going to be read has something called a promoter region. So it's the location on the DNA just before where a gene begins that says this is where RNA polymerase can bind on the DNA to do transcription. Okay. This site, this promotion location, the sequence is called a TATA box, which is this box right here. Okay. So it's the binding site for RNA polymerase, and I'll explain what transcription factors are in a minute. So RNA polymerase will bind to here and then always read the side that starts ATAT. Okay. The reason we call it a TATA box, though, remember that we're making the DNA 5 to 3. So the coding strand always says TATA, which is why this is called a TATA box. Okay. Transcription factors are usually things like steroid hormones that are going to help the RNA polymerase attach to the promoter site. Okay. The rest of the slide you don't need. It's AP material, so you can skip it. Okay. So the transcription factors plus RNA polymerase make up something called the initiation complex. Okay. So those three things bind to the promoter region. Okay. And it's going to trigger the binding of RNA polymerase to the DNA. So these hormones, these purple bubbles, are going to enhance the sticking of the RNA polymerase attachment to the promoter site so that transcription can begin. Okay? Then, in terms of matching bases, that's relatively straightforward. All you do is you use the base pairing, right? The only change is that A goes to U instead of A goes to T. Otherwise, C, G, T, A, U, G to C, T to A, C to G. And then A here will go to another U. So it's just base matching that way. Okay? Again, going from the 5 to the 3. Okay. Now, one thing about eukaryotic genes, and the term junk is a bit of a misnomer now. At the time, I mean, these slides are nearly 20 years old, right? And back then, people really didn't know what these in-between sequences of DNA were. They just thought there was junk. It's like bizarre pieces of DNA. No one knew why they were there. It turns out today we don't think they're junk, but they are regions of non-coding DNA. Okay. It turns out about only 1% of the genetic material in your DNA actually makes protein. The rest of it either is unknown function or is used to regulate various things inside your cells. So to turn genes on or off, things of that nature. So very little genetic material is actually used to make your proteins. Okay, so by eukaryotic genes have junk, I mean that eukaryotic genes are not continuous. In prokaryotes, if you have a section of gene then that whole gene is continuous. Start to end, you read the whole thing, and the whole thing is a gene. It'll make up one protein product. Not so in eukaryotes. In eukaryotes, you'll have bits in blue, right? This is a eukaryotic DNA. The bits in blue are going to make up the parts of a protein. Okay? So they're called exons, right? It's the actual gene. So expressed or coding DNA. The orange bits are something called introns. These are the non-coding bits of DNA. Okay? They're the in-between sequences. Before we can actually make a protein at this ribosome, we have to get the introns out. Okay? So there's a technique that eukaryotic cells do called mRNA splicing. Okay. So it's part of something called post-transcriptional processing. So after you've copied the DNA to RNA, you now have to do a little bit of processing. Okay? So the eukaryotic mRNA needs work after being made. Okay? So here's the original DNA. Okay? It's about 10,000 bases in length. Okay? When you make the pre-mRNA, it will also be about 10,000 bases in length. This is what we call the primary transcript. Okay? It has both the exons and the introns in it. Okay? What we have to do now is cut these orange bits out. So we need to edit out the introns. Okay? So when we remove these orange bits and take them out, you're going to get the shorter green sequence down here. This is the mRNA transcript, right? This is the mature transcript. So there's a eukaryotic DNA. There's the primary transcript, also known as the pre-mRNA. When you remove all the introns, you finally have the mature mRNA transcript that can be used to make up the protein. So you go from about 10,000 bases to about 1,000 bases when you get to the mature transcript. Okay? So that's quite a difference. Now, how does this actually occur? Load. Thank you. Genetics. Okay, so what I've been talking about up to this point is right here on page 321. Okay, let me zoom in a bit. So on page 321, it'll talk about post-transcriptional modifications. This idea of introns coming out is on page 321. Okay, the question is, how does the cell actually do it? Go to the next page and go to this diagram, figure 3 on page 322. Okay, so what do we got here? We've got, here's a section of pre-mRNA. That's exon 1. There's the intron that we want to cut out. There's exon 2. We also have something called, we have a combination of things called proteins, which is a purple blobby bit, plus something called a small nuclear, small ribonucleoproteins. Okay. So these small nuclear ribonucleoproteins plus these purple proteins get together and form something called a SNRP, okay, an SNRNP, okay. Several of these SNRPs will get together, okay, to form a structure called a spliceosome, okay. And what that spliceosome and SNRPs are going to do is they're going to force the intron to form a loop with itself. Okay. Once that loop is formed, this SNRP is going to cut one end of the intron. Okay. Which forces the intron to fold back on itself. Okay. And bonds to itself. Once that occurs, the SNRP will cut the other side and release the intron, which will be degraded. Okay. The two sides, the two exons, 1 and 2, will now be put together by the SNRP. The SNRP will let go and look for another intron to splice together. Okay. So this is what a spliceosome does. And spliceosomes are composed of a bunch of different SNRPs. Okay. So that's on page 322 of your textbook. The reason is because at the time I made this PowerPoint, we didn't know about how SNRPs existed, and I haven't really had the time to make a new slide for this yet. Hopefully I will this summer. But up to this point, then, I haven't, which is why I keep jumping to the textbook. Yeah, pretty much. Okay, so now the discovery of exons and introns was in 1977, Nobel Prize in 1993. Okay. Now, the thing about splicing, the splicing has to be accurate. Okay. There is no room for error at this point because you're trying to make a functioning protein. Okay. So a single base added or removed is going to throw off something called the reading frame. I'll explain the reading frame in a bit. Okay. This blue box highlights what's supposed to happen. Here's my original DNA sequence. Okay. In green are the introns. The red is in green are the exons. In red is the intron. And the blue is just to designate where the beginning and the end of the intron is. So we want the red bit out. Okay. Spliceosome comes and gets rid of this red section. So you're left with this. Okay. When we go to make a protein, we're going to read these nuclear bases in triplets. So we're going to break them into sequences of 3. And then each sequence of 3 will represent an amino acid. So in this case, this represents an amino acid sequence of methionine, arginine, serine, aspartame, lysine, glycine, and histidine. This is what's supposed to happen. Okay. But in the yellow box here, here's what happens if the splicing goes wrong. Okay. Same starting sequence. So the top lines are exactly the same. This time, one of the red G's got left behind. Okay. So we have one too many letters. So when we break them up into groups of 3, the sequence changes. The reading frame, right, the groupings of 3 has shifted. Because of that, the amino acid sequence changes. And not only that, we actually get a mutation that causes the amino acid sequence to stop prematurely. So you can't have an error occurring like this. Otherwise, right, you basically create a non-functioning protein. That's not to say that doesn't happen. It does. But it's pretty rare. Okay. Now, there's one small problem we have with the whole concept of the human genome. Okay. How many proteins would you estimate there are that you need to make in order to make a human being? How many proteins do you think you guys make? No, much less than that. It's under 500,000. Yeah, about 100,000 different proteins. Okay. The Human Genome Project was finished in 2001. How many genes do you think? So if we make 100,000 different proteins, according to one gene, one polypeptide, no. How many genes do we need to make 100,000 proteins? 100,000. When we completed the Human Genome Project and we actually counted, turns out there's about 20,000 genes in the human genome. So the question becomes, how do you get 20,000 genes to make 100,000 proteins? There are 5 times more proteins made than there are genes in your body. Okay. One of the ideas is this concept of alternative splicing. Okay. Perhaps it is possible that you can make alternate different mRNAs from the same gene. Okay. When is an intron not an intron? So assume that you've got this chunk of DNA. You've got 3 blue exons, a green one and a brown one. All the light blue stuff are the introns that you want to get rid of. Okay. So you make the RNA transcript. So the dark pink bit you want to keep. And then whether you keep the green or the brown depends on which mRNA you want to make. If you want to make gene 1, you go pink, pink, green, pink, and the brown becomes an intron and you get rid of it. But if you want to make gene number 2, then you go pink, pink, brown, pink, and green becomes an intron and you get rid of it. This way, from the same sequence of DNA, you can get multiple different mRNAs that could build a family of related proteins. And this may be how we get only 20,000 different genes making 5 times as many proteins in our bodies. Okay, so we treat different segments as exons. Okay, and so it means it gets really hard to start defining what a gene actually is. Okay, it's 1:20. I got to go make sure that my kids are back in school. So take a 15-minute break. We'll start again at 1:35. Okay. So there's a little bit more post-transcriptional processing that needs to be done. Okay. The other thing that we need to do is we have to protect the mRNA in the cytoplasm to make sure that it lasts long enough to give us the proteins that we want to make. Okay. So we have to protect the messenger RNA on its trip from the nucleus to the cytoplasm. The problem, as I stated before, is that enzymes can attack the messenger RNA. Now, the enzymes will never attack in the middle. They tend to attack at either end. So if you can protect the ends, you can make the messenger RNA signal last longer. So the way we do that is by two things. At the 5' end, we're going to add a guanine triphosphate cap. This basically acts a bit like a helmet and prevents enzymes or slows down the ability of enzymes to chew away at the 5' end. The other thing is we add what's called a poly A tail. So what we're basically doing is we're sticking 50 to 250 adenine nucleotides to the 3' end of the messenger RNA. That means that in order, before the signal gets destroyed, the enzymes have to chew through all these adenines first. The more adenines you have, the longer the signal lasts. And therefore you make more proteins. Okay. So that's transcription. Now we need to talk about translation. Okay. So going from nucleic acids to amino acids. Now we're switching languages, right? Because we're going from one type of macromolecule to a different type of macromolecule. So we have to make a switch. So this is like going from German to English. Okay. Here's the problem that we have in terms of coding for proteins. DNA has 4 bases. Okay. A, T, C, and G. Now, converting that to mRNA is pretty straightforward because mRNA also has 4 bases. So to get a 1-to-1 ratio between DNA and mRNA is pretty easy. I mean, the only difference you really have in terms of the bases are uracils for thymine. Otherwise, they're identical. Okay. The question is, how do we go from 4 messenger RNAs to 20 different proteins, amino acids, right? Because there are 20 different amino acids. How do you get 4 bases to represent 20 different amino acids? So I'm going to type this in the chat. If you had a 1-to-1 relationship, in other words, one base meant, yeah, if you had one base, okay, represent one amino acid, you'd only cover 4 amino acids. Okay? Insufficient. If you let pairs of bases, okay, so let's say AA equals 1, AC equals a second, AU equals equals a third, and AC equals a fourth amino acid. That gives us 16 combinations. Still insufficient because we've got 20 amino acids. So as Vedant said, if we use triplets, 3-letter codes, so we go AAA equals 1, AAU is the second one. AAC is the third one. And AAG equals the fourth one. You're going to get 64 different combinations. Okay. Then CCC, CCA, CCU, CCG, TTA, UUA, UUC, and so on and so on and so on. You're going to end up with 64 different possible combinations. And since there's only 20 amino acids, you've got more than enough combinations to cover the 20 amino acids that you have. In fact, you can now have redundancy, and therefore this gives you even more security. Okay, so this is how we do it. We use 3-letter combinations. Okay, so mRNA ends up coding for proteins in triplets. So here's my DNA. If I convert that to the mRNA, then mRNA is AUG, CGU, GUA, AAU, GCA, UGC, and C and GCC. Each 3-letter bit is called a codon. Okay, each codon represents a specific amino acid. So this sequence represents the protein methionine, arginine, valine, aspartine, alanine, cytosine, alanine. Okay, that's the amino acid that's basically made. That's the amino acid sequence for the protein, rather. Okay, so cracking the code was done in 1960 by Nirenberg and Korhana. Okay, Crick is the first person who postulated that there was a 3-letter triplet codon system. Okay, so why did the red bat eat the fat rat? Right, so that's a 3-letter system in English. So Nirenberg worked out 47 of them. Korhana worked out the remaining 17. Okay, and what they figured out was the mRNA to amino acid match. So the way they did it, they made up a bunch of fake mRNA or artificial mRNA and put it in a test tube with ribosomes, transfer RNAs, amino acids, everything you need to run the ribosomes. So when they created an artificial amino mRNA with only U's in it, so U, they got back phenylalanine as the amino acid sequence, which told them UUU has to mean phenylalanine. Okay? And they did this for all the different codons. Okay? So this is them getting the prize back then. So this is what the mRNA codon looks like. Yeah, you just take the different nucleotides and you stick them together. You take triphosphates, put in the enzymes of RNA polymerase, and you just get the polymerase to start stitching it together. You would have a template sequence of, yeah, you would probably make an artificial DNA first, and you could do it that way. So some of the things you should notice from this table, okay, this is the code for everything, okay? Now, notice the bases that are used. They contain U, not T. So what kind of nucleotides are they using here in this table? Yeah, codon tables are always RNA tables. They're never DNA tables. Okay, you will never see a codon table for DNA. You will never find one with thymine in it. It's always RNA. It's always uracil. Okay, this is probably one of the strongest pieces of evidence that we have for the common origin of all life, because it doesn't matter what biological life on this planet that you talk about, it uses the exact same sequence. Okay, the code is redundant. So there are many ways to spell the same amino acid. In fact, in many cases, you only need the first two bases, right? If you got an A and a C, it's pretty much going to be threonine. Doesn't really matter what the last letter is. Okay. If you have a Cu, it's leucine. If it's a G and a U, it's a valine. So you can pretty much determine the DNA code just based on the first two letters. The third letter simply gives you redundancy. Okay. We'll get there. Several codons exist for each amino acid, right? Leucine's really got a lot. It's got 6. Okay. Threonine has 4, valine has 4, alanine has 4, glycine has 4. Some of them only have 2. Arginine has 6, 4 here and 2 down here. Why is UC always one type and AG? Well, AG can be split in two. Yeah, I mean, that's just evolution.
Okay, like wait, I just mean wait, can you hear me?
Yeah.
Oh, yeah, I just mean like or
Is it
whenever.
always UC in the third base and AG in the third
Yeah.
base? Yeah, just dumb luck.
Okay. Like how U and C are always the same.
Yeah, no, it's just dumb luck. Okay. So this feature of the third base being flexible is called third base wobble. Okay. And the advantage of it is that the third base is the one that is usually prone to mutating. For Vedant's question about the UC and the AG, generally speaking, right, A and G are both the purines, which are double ringed, which is why they tend to pair up. UC are the pyrimidines, which are single ringed, and which is why they tend to pair up. Okay. But let's look at threonine. If ACU mutated to ACC, what's it do to the amino acid sequence? Nothing. And that's why third base wobble exists. You can have a mutation that does nothing. So you replace one base with the same base, or replace one amino acid with the same amino acid, and therefore has no impact on the actual phenotype of the organism. Okay. That's a big, big advantage. Okay. Now, Grant's question about AUG. Okay. AUG is codes for the amino acid methionine, but AUG is also something called the start codon. And what AUG does in about, I think it's between 95 to 99% of the genes in the human genome, AUG is the sequence you find at the beginning of the gene. It's like capitalizing the first letter of a sentence. It tells you where the gene begins. So whenever you're, for the most part, whenever you're starting a gene in a human, you're looking for the sequence AUG. Okay. You also have 3 sequences. Yeah, usually. UAA, UAG, and UGA. These are the stop codons. They're found at the end of a gene, and they're going to basically designate where the gene ends. So methionine, or a start codon, tells you where a gene begins, and these ones tell the ribosomes where to stop making the gene. That's the purpose of those 4 codons. Okay. All right. Now, something you need to watch.
codons. If you chose to watch this short video, you are perhaps searching for a little additional help to read a codon chart. And you've come to the right place. We're going to assume you already have the background from our DNA versus RNA video and our protein synthesis video. If not, you might want to check those out first, or otherwise jumping right into this isn't going to make much sense. Oh, and in addition, we have a video companion handout to this video, which can be helpful. Expand this video's details for the link, as you may want to download it first. So if you had this DNA template here, you'd have this mRNA built. I know this because of how RNA bases pair with DNA bases. Notice how these mRNA bases are written here in groups of 3? Each is a codon. Bases are read in threes, so a codon is how these bases will be read. In this example, there are a total of 5 codons, 15 individual RNA bases. By reading a codon chart, you can determine the amino acids that will be brought in by the tRNAs. You can determine the sequence of amino acids that make up a protein. And if you're wondering why this matters, understanding how amino acids are coded for helps us understand human traits and how changes in amino acids have the potential to alter those traits. Not just for humans either, but all living organisms. The codons and their corresponding amino acids have been discovered by scientists. There are many ways you can represent the information in a chart. The most common ways are the rectangular codon chart and the circular codon chart. Let's get started. The first codon. Remember, codons have 3 bases, and codons are on the mRNA, is AUG. That means the tRNA anticodon will be UAC, and we know that because of the base pairing rules. Now, which amino acid will that tRNA carry? Time to use the codon chart. We'll use a rectangular one, and remember, it's the mRNA codon that we look at, not the tRNA anticodon. So notice on the left side of the chart, it is for the first base. The top is for the second base, and the right side is for the third base. We're looking at the codon AUG. We go in order. First base letter, A. We isolate this row here and only this row. Second base letter, U. We isolate this column here and only this column. We've narrowed it down to this area here where they intersect. Third base letter, G. Here. The codon AUG codes for the amino acid methionine. Fun fact, methionine is commonly the starting amino acid for many polypeptide chains, as AUG is a start codon. Next codon is CCA. That means the anticodon for the tRNA will be GGU. Now, what amino acid will that tRNA carry? Time to use the codon chart. This time, we're going to ask you to pause the video while you try to determine which amino acid this tRNA would be carrying. Again, don't forget to use the mRNA codon.
Oh, what's the amino acid then? It's CCA is the codon, so what's the amino acid based on the table? Yeah, C, this row, C, this column, and then the A right here, proline. Okay. Okay, so I wouldn't worry about the circular charts that they kind of were mentioning, as long as you know how to use this rectangular chart, because this is the kind of chart I will always give you. I have never in my life seen a circular chart. Okay. And I've been doing this for 20 plus years. I'm sure they exist, but I've never seen one that's in a circle. Okay. But these rectangular ones are really, really common. So let's try a few. What amino acid does AGU give you? Let me present. I guess I have to present you the chart. You don't have your textbook open. Here we go. So AGU. Okay. GUU. Yeah, that's down here. UGU. Yeah, up top here. Right? So find the first base, then the column, and then the individual one. Okay. If I give it to you as a 3-letter code, just give me the 3-letter code. If I give you the table with the full name, give me the full name. Okay. All right. So methionine is special, as are the 3 stop codons. Okay. We've already discussed why wobble is good. Okay. So how are the codons matched to the amino acids? All right. Well, there's the DNA, right? We can go to the mRNA, which are the codons. Okay. Well, what brings the proper amino acid in is something called a transfer RNA. So transfer RNAs are attached to the correct amino acid, to their amino acid, and the transfer RNAs are going to bring in the appropriate amino acid to the mRNA and the ribosome in order to bring the two sides together. How does the cell check? Okay, the mRNA has 3-letter codons. On the transfer RNAs, the transfer RNAs will also have a 3-letter code on them, but it is the complement to the mRNA. So if the mRNA says AUG, the transfer RNA has to say its opposite, UAC. This is called the anticodon. Okay, so the mRNA has the codon, and the transfer RNA with the matching anticodon will bring in the correct amino acid. So AUG, the transfer RNA has to be UAC, and it'll be carrying the amino acid methionine. If it's CGU codon, the anticodon would be GCA, which is arginine. But remember, when you're looking at this codon table, this codon table goes directly from the mRNA codon to the amino acids. It skips the anticodon. So you don't need to figure out the anticodon for the transfer RNA in order to figure out the amino acid sequence. I can ask you what the anticodon would be if I give you the codon, but you don't need it to figure out what the amino acid is going to be, because this does the translation for you. Okay? That's the bit that gets people. So that little truck represents the transfer RNA. Okay. So what I've talked about up to this point is the theoretical side. How does the mRNA turn into protein? Well, we're going to be using anticodons and we're going to be using amino acid sequences. But what actually happens is the next bit that we need to cover. Okay. Now, let me just check something. Classroom. Is there anything helpful here? Transcript translation. She's not at work. She's at a doctor's appointment. Huh? Because she never hears it. Okay. I'm just checking something right now. No, go away. Okay, so what I'm going to do is I'm going to show you the first part of this.
Our protein in the next.
I'm going to show you the first part of this Amoeba Sisters up to the end of transcription. Yeah, probably. Let me look at the date. So we're probably looking next week sometime. Not quite sure exactly when yet. We've got how many left? Seagate backup. No, in the unit. Genetics. Mutations, operons. Yeah, 2 more PowerPoints. So we'll probably finish up this week. And then, so yeah. And plus a gizmo. So we'll be done the unit by the 4th. So sometime next week will be the test. Probably, let's take a look at June. 9th, 10th, something along those lines. We're gonna finish. We're gonna finish this early, as early as we possibly can. Because I still have 2 more units to go through, and they're gonna be compressed because we're short on time. I have 2 weeks, 1, 2, 3, 4 days to complete this course. Yeah, not 3 weeks. Because we're supposed to be done on the 18th, so that you guys have time to finish handing stuff in. So you guys basically get 3 support days at the end of the term to hand stuff in and such and work on things and improve marks. So the goal is to be done all new teaching by the 18th is what admin asked us to do. So we're getting close. All right, so let's watch this protein synthesis bit.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA. in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart.
Okay. We'll basically stop there because the rest of it goes into more details of translation, which we would rather cover tomorrow in class. Okay, so it's 2:16. I would strongly suggest you review what we went over today, and I will see you tomorrow. So take care.